The Institute for the Study of Peak States presents the first International Psycho-immunology and Psychobiology Research Symposium. Psycho-immunology for viral and bacterial infections with implications for coronavirus. Presented by Kirsten Lykkegaard.
Our next speaker is Dr. Kirsten Lykkegaard. She is the co-director of research of the Institute for the Study of Peak States. She's a doctor of veterinary medicine, holds a PhD in pharmacology, and has 17 years experience in medical research focusing on appetite regulation, obesity, and type 2 diabetes. Over to you, Kirsten.
Thank you, and thanks for being with us today. I'll briefly go through some limitations of the discussion and some safety precautions. So what we're sharing in this presentation is limited due to the safety concern, which holds the risk of disturbing homeostasis of the pathogens, which can cascade into severe problems. So we're not going to go into depth on some of the research that I will be mentioning, and also do not experiment on yourself with any of the material in this presentation. Thanks. So I'll move on to the extrapolations that we've made so far on pathogens in general. So our belief is that bacterial and viral genetic material is foreign and should not be inside our cells. Bacteria and viruses exploit early developmental events. That means the person unconsciously wants the infection if you have trauma in these events. And that unconsciously makes us perpetrators and not victims of all these diseases. You can have natural immunity to infections because of trauma not existing in certain developmental events. You can have them on a case-by-case basis, or you can be so lucky, which is very rare, that you have a general immunity to bacterial and viral, and that is a peak state. This is what I'm going to go through. So yesterday you heard about the fungal diseases of OCD. I'm going to just touch base on the principle of the voices. Again, this is a fungal that has not been identified. The voices is a full working process that we offer, call it silent mind technique. And the OCD is in the middle phase of testing, like Jennifer explained yesterday. Then there's the bacteria disease of Lyme, which we've had several test cases on. I'm going to just go through an example of that. And then for the virus parts, we have had one test case on shingles, which is caused by the virus that causes chickenpox. And then I'm going to end up talking about the COVID-19, which is caused by the severe acute respiratory syndrome, coronavirus 2. And there we have one non-confirmed COVID-19 test case. That was what we could dust up. How to say that? We could not find one that was confirmed to work on so far. So just a brief summary of how the psychoimmunology works. We actually work with the pre-birth developmental event because that is what the pathogen exploits, our unconscious desires that come from these events. So just imagine a pre-birth developmental event that could be anywhere from ovulation, conception, implantation, those big events where things really change in our biology. In those events, what we've seen, and that is with both peak states induction, but it's also where we get vulnerability to diseases. It is set up by trauma. So you have the event and it doesn't quite go right. And so in the past event, or even in the present, some of the diseases is something in the present that gets triggered that then gets your awareness to that early event where the trauma was set up. So you reach out to your surroundings for help. And you do this for one of several reasons. One, it's like you want a substitute for something that you missed getting in this event. And that's a physical, I mean, if you should think of this as a biological thing that's happening in this event. So something like physically, chemically is going to change in your body, and it's not quite right. So you're missing something, and Lyme disease and OCD are two of those examples. Also, you want something to help you avoid facing a trauma in an injury situation. That could be, for example, Asperger that Gaetan was talking about yesterday. It's like something is happening to you, and you try to help yourself out in that early developmental event, and in that you get vulnerable to this. It could be a bacteria, could be a virus or anything like that, but in Asperger it's a bacteria actually. Also, it can be you associate survival with the presence of a pathogen or a contaminant. I give an example of schizophrenic voices. There's also mercury. And then the last one is that there can be interactions between people that can trigger primary cell damage by pathogens. And examples of that is autism and psychosis and schizophrenia. Secondly, what we figured out, so this is the last slide was the developmental event setup for it. And what we found out was that these pathogen types has its own characteristic emotional tone. So if you actually have a client feel into that pathogen in the present, there is something going on in that pathogen that irradiates an emotional tone. Also, that pathogen can release a fluid, or it has some kind of characteristic fluid environment that fools the immune system. And so we can actually divide our techniques, and they all sort of works. They all have their limitations. So the first generational techniques that we developed, they heal in that developmental event that I talked about in the prior slide. This works, but for some people, the traumas can be too difficult to face. I'd say that we do have we have some really good processes. Silent mind technique and Asperger actually work in this first generation manner. And they work really well. Second generation techniques, they use body associations and generational trauma healing on the emotional tone of the pathogens. Here, we've only done that with the virus, and I'll show you how it works on the Shingles case that we have had. So we think it's too unreliable in the long run, because identifying and facing that emotional tone in the pathogen is not easy. So third generation is what we're keeping our hopes to, is that they focus on healing the person's unconscious craving for this taste of the fluid in the pathogen. And at this point, this is also very much easier technique for people to do you can actually do in the present. So, and facing the fluid is a lot easier. So let me go through this one, which is the ribosomal voice. That's what we call the most common form of voice that you can have, which is caused by fungal disease. And these set up trauma in utero. And in this case that you see over to the right here, there is the fetus inside. There is some sort of physical trauma. The fetus has this trauma and reaches out to its mom. And in this moment, the mom is sad, for example. That means the fetal will couple survival, because this is a trauma, but the fetal survives. It'll couple these two things, sadness and survival. So a body association is formed, and later there is this interaction. There's a common infection that we all have inside our cells, and this is the fungal that we assume is causing this problem. So this fungal will interact with the ribosome, and that actually sets up the whole problem. You can have associations without this infection inside the ribosome, but at this stage, it's like a muted voice at this point. and it causes just thoughts of mind chatter. And thoughts, it's not the thought I need to remember buying milk. It's more like thoughts in the background that you can't shut off. So, and it can actually be like a little refrigerator noise in the background. Later on in life, if you encounter a really bad survival trauma, this can activate that change that happened due to the fungal, and then it sets off this problem with the voice being loud. And now the voice actually contains the emotional tone of whatever was coupled with survival. So if mom felt sad, that would be a sad voice. And we can eliminate those either one by one, which isn't a sort of immunity, but that's getting rid of. But we also have the overall technique with which is called silent mind technique that actually gives immunity to the fungal. It actually goes back to, like I said, it's a first generational technique. So it uses the developmental event when this infection happened of the fungal. OCD, Jennifer mentioned that one yesterday. And she also said that there are two different developmental events. They're actually pretty closely related, but still separated. So there are two fungal, one in one event, one in another. And however, the reason for wanting in both events happens even earlier. And that is very convenient because then we can use that earlier developmental event to give immunity. So healing all the traumas in the very early developmental event fixes the need for that. It's a substitute if you think about it. In those two events, there is something that is set up by the first event that makes you reach out for this fungal issue. And so we can heal by first generational technique to give immunity. That works, like Jennifer gave the two cases that she'd worked with and how well that actually improved their symptoms. We are now going back to see if we can refine this technique to be more of a third generation technique. So the two funguses is the fear fungus, and which is caused by the first event, causing anxiety symptoms, and the second event contributes to contamination symptoms, which is why we've named them fear fungus and contamination fungus. So that looks very promising, that we can get rid of that. Then we have Lyme disease. So now we go into something that actually causes the disease. That's caused by Borrelia, and it's carried by the ticks that you can get while you're out walking in the forest. And that bacteria goes into your body, and it can if you take antibiotics up front, you can get rid of it, but up to 10 to 30% of patients do actually have symptoms that last for a chronic period of time with severe fatigue, especially the pain joint. The joint pain is a problem, and that was what I showed on the slide yesterday. Can also be misinterpreted as rheumatism. So what we've done in this case is that, let me first tell you that actually the reason why long-term, the chronic symptoms here it's funny because now it works over here. So in these cases of chronic, not infection, but the chronic symptoms, so where the Lyme bacteria is actually still present in the body but do not respond to antibiotics, that is very likely due to that they actually come in different forms. So if you look at these are just a little image of, they're formed like little corkscrews, these bacteria. So they can be sitting in like the shoulder joint or the knee joint, but they also kind of put themselves into a stage of where they're outside the immune system. It's called a biofilm and round bodies. And it's almost like a little depot of bacteria in the body. What we have noticed in these, the clients that we worked with, noticed that these spirochetes gives off a liquid. And that liquid is actually something that is similar to a liquid that appeared in an early developmental event where the trauma happened. And so now in the present, when this bacteria comes into the body goes, oh, this is what I needed in this old trauma moment. And that's what we call addiction in this case. So the body actually goes unconsciously hanging on to it, even though the disease will cause more harm to the person than fixing that trauma moment in the past. So there is the body won't let go, but the body is damaged by what's going on. So what the simple version, we've tried actually, I think all 3 generational techniques on this, but this is where this is almost a 3rd version generational technique where we looked at the liquid and had the person get rid of the, you can say, addiction to the liquid. And that actually removed symptoms and the spirochetes feeling in this person's body. But still that sac-looking thing in the body there, we're not sure if we got rid of that, but the symptoms went away. So there's good promise for this technique, and we need to keep working on it. Then the next one is very, I think, very, it was an awesome session, this one. So this is actually where it started for me, was that I had a client that had depression, but also the client was suffering from shingles, which is the the chicken pox that most people have had as kids. The chicken pox virus, which is varicella zoster, actually resides inside the nerve cells around the spinal cord and the brain, and can get reactivated, just like you can have a herpes, it's the herpes family. You can have a cold sore that is also a permanent infection or latent in the nerve system. So this client had, first we were actually just focusing on the depression in this client. And the symptoms of the depression was this heavy feeling in the chest, like a big lump. And it was like, it's so hard to do something in the world, and I really don't care, and I don't give a shit. That was the feeling in the chest. But before we actually worked on that, we also felt there was something in the back, but that was probably due to the shingles. So first we fixed this thing here. And then after that actually healed, and the client felt that it was like leaving through the eyes, almost like up through the head. And it was such a relief. And then with the shingles on the back, there was just a radiation of loneliness. And so we healed that. This is where we think this technique, it works, but it's very hard. You need a really good client to be able to feel what is the emotional tone, because this is the emotional tone technique of what the virus feels like. So there was a radiation of loneliness, and under that there was a radiation of anger. And as we were healing that, and it was gone, the client was just, it was like that shingle and the pain and the itchiness, everything just went away in one go. And it took about 10-15 minutes. And this was where I knew something is going on here that we could actually proceed on to work on as a viral thing. And I thought this was pretty interesting, because this was something Grant wrote in 2016, and Grant talked about it yesterday, in a sense, because he was saying that in these days where we have superbugs that are starting to proliferate, antibiotics and antifungals rapidly become less and less effective, and our model does predict that we can actually make people immune to entire classes of diseases. And this is actually what I described in the first place, that there are people who actually don't get sick. You don't hear about them. They probably don't even make a big fuss about the whole thing. Very rare. So what Grant wrote here that was that we don't want to wait until worldwide pandemic sweeps our species. And I guess we've been busy doing other things, so I guess we we are in that situation right now. So that's why I will go into the next few slides, and it is very preliminary work. So please take that into your consideration. So just very briefly, this is a linear single-stranded positive RNA genome. That means this virus does not need to go into the nucleus of our cells to replicate. So it consists of proteins, RNA materials surrounded by protein and then encapsulated in a lipid layer here. It's a very sort of small virus, to 27,000 to 30,000 bases, and it binds to our receptor for angiotensin converting enzyme 2, which is on the epithelial cells of lungs and our gastrointestinal tract. And this one I thought was interesting, just to mention the virus outbreaks that have been in the world. You know the SARS that was in China, which is again a severe acute respiratory syndrome coronavirus. So this is the number 1. This is why they call this number 2, because it gives the same kind of symptoms in the respiratory tract. And then back in 2012, there was actually the Middle East respiratory syndrome related coronavirus. So these are all in the same family. But this one comes from the camel, and we think this one is from a bat. And I think this is I don't remember the animal, but so it's transferred from animals. They also have found, here I'm referring to this article down here, that they found that there are 2 types, the L and the S type. It relates to the proteins in the virus. And the L type is more prevalent than the S type. But the S type seems to be the one that was in the original one, and the L type is actually a little bit more aggressive than the S type. They think they have eliminated the L type in China, so it's the S type that is going around the world right now. Also, I'll just show you this one from this article that shows that it remains in aerosols. That means in the air for up to 3 hours is what they found here. They measured 3 hours. So you see that's how it decays or falls away. And on surfaces, it says here that it's more stable on plastic stainless steel than on copper and cardboard. So you see cardboard, it vanishes quicker than it does on the other 2. And the reason I mention that is just to show that sorry, that's next slide. This is just a slide showing how it binds to the receptor. It internalizes into the cell where it will open its envelope. It'll start immediately going to the ribosomal to read its RNA into protein. It'll make new viruses that then come out the cell. And so in a person, we have worked with one test case that we were so lucky, you can say, to be able to work with one of our colleagues who was infected with what looked like coronavirus. He was non-confirmed though, so this was the best we could work with at this time. So we found in him, by actually having the regression moment back to when the infection started, and to take a look at what it looked like. And there was aerosols in the air. This can come in through the airways. There were also some from shaking the hands of another person. And what was surprising was that in this aerosol, the droplets, there was just a goo feel in the air there, and those droplets had an amoeboid shape to them, even with a nucleus in the middle. And that's not reported in the literature at all. We don't know what this is, but what we picked up on was that it had a specific sort of taste to it, a feel to it. So, and this one had the same type of amoeboid goo, but it had a different taste in there. So what we've done was we tested in the first round here, we tested immunity to actually getting infected. So in this very early moment of this, of our colleague here, to see if the infection could be eliminated, in a sense that it didn't happen. So we used some techniques on the taste of these, so the taste of the goo. But this technique did not get rid of the virus. He was already infected, and we needed to go to a deeper level. So what we did was we looked at his lungs, and he had this feeling of this heaviness in the bottom, and it was a really black and dark and gooey feeling. And in there, we were able to pick up the emotional tone of the virus, and also the taste of the liquid that the virus was in. And so we've done some healing work on that, and that last approach seemed to be more effective. We have the hopes that this way of doing it to work with a client who's willing to do this process, who is confirmed COVID-19. So that's actually all I have, and I think that was. So we have definitely time for questions on this session here.
Okay, so the first question, Kirsten, was how has the determination of an intercellular fungus been made? How did a fungus get into a cell in the first place? This is from Patricia Shaw. So
the fungus that comes into it's in the, so if you think about the biology that you created inside your mom, and actually also inside your grandma, that they're already infected. So it's an opportunity. It's an opportunistic thing that happens. So these parasites are there, and if trauma goes wrong, which is typically generational, so it's like you're almost doomed to get these. They're part of our biology. I think you can say they've just settled into our beings.
Okay, thank you. Then the next question is from Dr. Douglas Schneider. I can understand the threat of trauma opening the fetus to the experience of the mother's sadness at that time, but I don't understand the connection between this experience of an emotional state and an intracellular fungus.
It's a bit complicated, and probably not for this talk, but there is a level of something going on that is, it's a layer. So first you get set up with trauma that links the emotional tone in your mom in that trauma moment to your own survival. So now you will actually, in a sense, want to surround yourself with that emotion, that sadness. So you will be looking in the outer world for that sadness. And it's actually part of that looking that triggers that fungal infection to do harm to your ribosome. So it's indirect, you can say.
Okay, thank you. Then we've got from Holly Rankin, how is it determined that it is a fungus attacking and affecting the patient rather than something else?
Well, that's where I don't know if the person asking this question listened to my talk yesterday about conflicting worldviews. I would love to, nail whatever that is. We have, when we work with these things, we definitely can classify what that we are working with feels like, and there we have definitely categorized that there are pathogens that feel in a certain way, and whatever that is, I would love to know. Yeah. So we're calling it a fungus in this sense, and it definitely, it has the feel of something that has that fungal feel. So.
Okay, thanks. Then we've got a question from Matthew Learning. What barriers exist that we have to overcome in order to have 10 test subjects who do have the coronavirus whom we can try this work on?
Well, I would be happy to have one in the first place. So what barriers do we have to overcome? We are a small team, so we can work with one at a time, I'd say. And that's what we would do, moving forward. I don't think that there is a barrier except time for the 10.
And maybe the question is, are we ready for that yet, or what else needs to happen before we're ready to be tested?
We're ready to do testing in a person who's who's willing to be working experimentally. Yeah.
Okay.
So maybe I mentioned that we all work voluntarily, and that our time is what it is. There's 24 hours in a day.
Yeah.
Yeah.
Okay. Then from Ilya Zakharov. So it's a bit of a statement and then a question, I think. So laboratory aerosol and real air are two different things. As with dead RNA on the surfaces and minimal probability to catch the infection while touching things. What are the exact emotional tones and the taste of liquids?
Well, that was the reason why that wasn't on the slide, I would say, at this point. We want to make sure that what we release when we release it is working, and it's the exact feelings that are there. Because they matter. I mean, when I did the shingle client, it really mattered. You need to nail that, and that's why it's always a difficult thing, and why it's not a process that can work like a charm every time. At this moment, anyway, we need more testing.
So I've got a question from Amy Zook. So is a biographical or generational regression technique used for the COVID virus?
We haven't seen biographical so far. So it seems like these infection patterns are set up both by the traumas that you experience inside, but they are driven mostly generational, I'd say.
Then a question from Evan Fee. What healing technique did you use for the COVID-19 virus? Body association technique, ? Can lay people use the description of the technique in the Voices book? Sorry, they're two different questions. So you want to answer the first one around what was the healing technique for the COVID-19 virus?
So right now we're looking for this third generation is body associations technique and generational, and we're also using other to clean up. So it's not only. So right now it's a matter of finding out how to do this simple, but do it so that it actually works. So I can't say for sure what we'll end up with. And for the voices, what was the question again? It was, I think it's actually published in Silence the Voices book, how to eliminate the voices one by one. If you want the full silent mind process, which actually, because of this fungal also does other things besides doing the damage in the ribosomes there, there's more to gain by the full process of immunity. And that's not released in the books.
So I've got a question following up to having test clients who are confirmed COVID-19. Is there any location restrictions on that? Does it matter where they live?
No, it would be on Skype.
From Kate Dole, to strengthen the immune system, is it a common process for everyone or does it vary for individually?
To strengthen the immune system, we don't know how to do that as such. We have an idea of what causes weaknesses maybe in the immune system. So we have a ton of different things that we could research. So I don't know how to answer that question right now.
Okay.
Our approach is not strengthening it. Our approach is to get rid of the desire for the unconsciously desire for having the infection.
Did you mention an immunity peak state? Because I've got a question from Evan Fee. Can you speak more about their immunity peak state?
So in this slide, Grant actually is stating that our model predicts that we can actually make people immune to entire classes of diseases such as, and then also, I need to minimize something here. Although exceedingly rare, there are people who are exhibit these kind of immunities. So it is true that some people out there are in totally different states than the rest of the normal distribution curve. And our model predicts it. We don't know how to induce it yet. I mean, we're working in these areas of the developmental events where probably some of these diseases are set up. So future could bring some of more clarity on that whole area.
Okay, thanks. I guess just adding to that, Lisa Gorland said, an historical example of the immunity peak state, the explorer Percy Harrison Fawcett, who explored and mapped the Amazon in the early 20th century. So, like you were saying, there are a few individuals with that state. So again, following up on that same one from Dr. Mary Pelletier, do we know the key event or events for global immunity?
I guess we don't know them all. We think we know some of it, yeah.
Yep. Okay, I've got a question here from Sergio Lil. What else does your research say about psychosis and schizophrenia outside of schizophrenic voices part you guys have already figured out?
So that's very early stages of that research right now, but it does incline that there is a pathogen involved in the negative emotion, effect of schizophrenia. So the ones that is not cured by, some schizophrenic are diagnosed based on their voices, and that's what's driving their the whole disease spectrum, you can say, their fear or whatever they're having. But the rest of symptoms are not cleared with the voices. So we think we have a window into what's going on there. And it's too early to talk more about it at this stage.
That's great. It doesn't look like we have any more questions. So thank you very much, Kirsten. The only thing that I would say, that as Kirsten has been talking about, this is very, very early days in the coronavirus research. I guess people might be interested in why we've all of a sudden started focusing on this. Obviously, with the current world situation, this was not something we planned on doing, but in alignment with our core purpose as an organisation, which is to find ways to heal humanity, we kind of felt compelled that we think our techniques, from a model perspective, it's theoretically possible for us to come up with a treatment for this, and so we thought we would do everything we can to actually help with that. So if we were able to do that, if we did come up with a treatment that looks like it actually does work, then our goal would be to work out how do we deliver this in a safe and effective way to get it out to as many people as possible as a philanthropic humanitarian project. So fingers crossed that the breakthroughs the research team are making do end up being successful. I think that's it for now. So thank you very much, Kirsten.
Thank you for watching. For more information, please visit peakstates.com.